What are the three stages of cellular respiration? The three stages of cellular respiration are glycolysis, citric acid cycle, and oxidative phosphorylation. Uh, what is the stage between glycolysis and citric acid cycle? Well, there is nothing as you can see here, but no, it's not true. There is a stage between glycolysis and a citric acid cycle. In fact, it should be called a step. So if there is a question on the test, if and it is asking what is the step between glycolysis and citric acid cycle, make sure to remember that it is pyruvate oxidation, like oxidation, Tion. So that is the step between glycolysis and citric acid cycle. What is glycolysis? Breakdown of sugar into two molecules, and each one is called pyruvate. Citric acid cycle is the one which will completely break down glucose into carbon dioxide and water. What happens in oxidative phosphorylation, which is the third stage or the last stage of cellular respiration, is that whatever electron carriers have formed in these three stages above, glycolysis, pyruvate oxidation, and citric acid cycle, they will be used for synthesis of ATP in oxidative phosphorylation. So glucose has been broken down. Glucose got exhausted by the end of citric acid cycle. So remember the three names of the stages of cellular respiration. We are going to talk in depth now about each one. So at this point, I would pause and simply memorize all the stages of cellular respiration. So if you're a visual person, here you go. These are the stages of cellular respiration. Don't forget, there is pyruvate oxidation here. Pyruvate oxidation. So between citric acid cycle and glycolysis, there is a step called pyruvate oxidation. Where does it take place? It very much takes place inside the mitochondria. So it is pyruvate oxidation and it takes place inside the mitochondria. Then where does glycolysis take place? Glycolysis takes place in the cytosol. And where does glycolysis take place? In bacteria. Yes, it is in its cytosol as well. But bacteria do not have citric acid cycle and oxidative phosphorylation. Is it true? No, bacteria do not have mitochondria. So bacteria also breaks down food to release energy and make ATP through all these four stages, and they all take place in the bacterial cell. Whatever takes place in the cytoplasm in eukaryotic cell takes place in the cytosol of the prokaryotic cell as well, and whatever takes place in the mitochondria will take place in the membranes or the intermembrane space, the space between the membrane and the cell wall of the bacteria. So try to remember that. I hope you memorized all the stages of cellular respiration. If they ask you what are the three stages, you will say glycolysis, citric acid cycle, oxidative phosphorylation. If they ask you what is the stage between or the step between glycolysis and citric acid cycle, make sure you remember pyruvate oxidation. So remember the order. The order is very important. GPCO, glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation. Let's take it a little further. Oxidative phosphorylation has two substages, electron transport chain and chemiosmosis. Remember, these are the two substages of the last stage called oxidative phosphorylation. Remember that glycolysis is the first stage and it takes place in the cytosol, and everything else in a eukaryotic cell will take place in the mitochondria. If it was a bacterial cell, then they all take place inside the bacterial cell because they do not have mitochondria. Okay, going to the next part of the chapter. What are the molecules which take electrons from when electrons are extracted or stripped off the molecules or the food molecules. They are called electron carriers. One of them is NADH. NADH is an electron carrier. NAD plus is the one which accepts the electron from food, and it will also accept hydrogen ions, and then it will become NADH. NADH will not keep the electrons forever, it will give the electrons away. So try and memorize or try to apply the logic of Leo says Gur, and remember that NAD plus is an oxidizing agent. NAD plus is undergoing a reduction because it is gaining electrons. Remember, Gur, gaining of electrons is reduction. NADH has electrons, so it is going to lose electrons. So when it loses electrons, then it is called reducing agent. And because it is going to lose electrons, losing electrons is oxidation. So NADH undergoes oxidation, or NADH is oxidized to NAD plus. It is not oxidized to NADH. NADH is oxidized to NAD plus. It is NAD plus. It should not be NADH. It should be oxidized to NAD plus. That is how NAD plus is recycled. You know that NAD plus is actually a B vitamin. Well, NADH will pass on the electrons to the proteins in the inner membrane of the mitochondria or the cell membrane of the bacteria. And when electrons jump like this from less electronegative molecule to more electronegative molecule, gradually more electronegative molecule or complex, then these electrons are releasing energy, and this energy is captured to synthesize ATP. And this capturing of energy is a process, and that will be explained later. That is what I'm going to talk about in oxidative phosphorylation. But remember that oxygen is a molecule which is the most electronegative in the cell, in the aerobically respiring organisms, and oxygen is driving this process. If there is no oxygen, this entire process stops. When oxygen accepts the electrons, it will also accept hydrogen ions, and water molecules are made. And this energy which is released by jumping electrons is captured to synthesize ATP. And ATP are synthesized in the sense ADP, which is adenosine diphosphate, is added one more phosphate. That is how it becomes ATP. Do not think that ATP is synthesized all new from the food. It is not coming off of food. No, ATPs are regenerated. ATPs are regenerated in the sense ATP is broken down to ADP and phosphate, and it releases energy for our work. This ADP and phosphate, whenever we provide energy through this electron jumping process, that energy is captured to put these two things together. That is how ATP is regenerated. Remember, the word is ATP is regenerated during cellular respiration. Let's go and find out what all you need for extra credit for glycolysis, and what all you need to remember. So let me pause here a little quick to explain something. Okay, so in glycolysis, I'm going to discuss what you need for extra credit. I would like you to draw this diagram, simplified model of glycolysis. Understand that these diagrams represent the inputs and outputs. Drawing is a learning. By drawing, you learn. Especially the visual learners, they learn very easily lots of facts in short time by looking at the pictures like this. And those who are not, you're putting in the effort to draw the arrows. So when you're drawing the arrows, remember that the uh, starting of the arrow, or the blunt end of the arrow, indicates that the picture or the word that you write on the side of the blunt end is something that is going into the pathway or into the reaction, and whatever you write at the tip of the arrowhead, they are the ones which are coming out of the pathway or the reaction. So glycolysis is a pathway. It is made of 10 reactions which are happening consequently. All of them summarized, they have given you the inputs. One glucose molecule, 2 NAD plus, 2 ADP, and 2 phosphates will make 2 pyruvates, 2 NADH, and 2 H plus, and finally 2 ATP. As you can see, there is no oxygen involved, there is no carbon dioxide formed here, right? So if you need to watch what happens in glycolysis and learn more details, here is the website. But you do not need most of it. All you need is this part which is coming up. Glycolysis has two phases: energy investment phase, wherein glucose is going to start to break down when the cell uses ATP molecules as activation energy. And then energy payoff phase is when glucose has started to break down and energy released is captured and stored into ATP molecules. So remember, glycolysis is breaking of one glucose into pyruvates. Yes, so if it is not glucose, if it is protein, amino acid, then it is not going to undergo the very first step of glycolysis. So it does not begin at the very first step of glycolysis. It will start its path somewhere in the cellular respiration. Well, let's stick to glucose for now. Glycolysis has two phases: energy investment phase, where 2 ATP are used to activate the glucose to start breaking down, and during the energy payoff phase, 4 ATP are formed. The question asks, what is the total number of ATP formed during glycolysis? You would say 4. If they ask you what is the total number of net ATP formed, if they use the word net, then you're going to say it is actually 2, because 2 have been used and 4 are formed, but finally what you get as net output is 2 ATP. Are there any other profits from glycolysis? Yes, the high energy electrons have been donated to NAD plus, which will be used for regenerating ATP during the last stage of cellular respiration. So yes. Coming to energy investment phase, as you can see, there are 5 steps happening consequently, one after another. Sequentially, one after another, and each step has its own enzyme. Good news, you don't have to memorize all the names of these molecules, inputs and outputs, and the names of the enzymes. But if this pathway is given, you should be able to say what are the inputs of step number 3 and what are the outputs of step number 3, right? Inputs are ATP and fructose 6 phosphate, whereas outputs are ADP and fructose 1,6-bisphosphate. Is energy used up in this process? Yes, very much, because this is an energy investment phase. Just like your business, you have to invest money at the very beginning and then you will make profits. Energy payoff phase has 5 more steps which happen after the first 5 steps. But here, see, energy is released from food and ATPs are made. So what is the net ATP production of ATP during glycolysis? It's only 2. What is the total number of ATPs formed during glycolysis? It is 4. If they ask you any other profits other than ATP, so yes, you have to say 2 NADH. Yes, the numbers are important. One glucose undergoes glycolysis to make 2 ATP, 2 NADH, and 2 pyruvates. That is what is given in this summary summary drawing, and you have to draw this for extra credit. There are more drawings coming up like this. Second step, or the intermediate step between glycolysis and citric acid cycle are the intermediate step is pyruvate oxidation. If there is oxygen in a cell, whether it is bacteria or eukaryotic cell, then the pyruvate is going to be oxidized further and going to participate in citric acid cycle. If there is no oxygen, then pyruvate will not be drawn into mitochondria or will not be taken through the citric acid cycle or pyruvate oxidation, and pyruvate will be undergoing fermentation. Does fermentation take place only in bacteria? No, it's not true. Fermentation can take place in our muscle cells. Fermentation does not occur in our brain cells, but yes, when our muscle cells are working too hard and you're exhausted and they need to still continue making energy, then they convert this pyruvate, which is the product of glycolysis, into lactic acid, also called lactate. And if your muscle cells have enough oxygen, then they are going to convert pyruvate into acetyl CoA in the mitochondria. So remember this, that pyruvate is a fork between fermentation and aerobic respiration. If there is oxidation, if there is oxygen in a cell, then it will undergo pyruvate oxidation. If there is no oxygen in the cell, then the pyruvate molecule will undergo fermentation. If it is yeast or bacteria, then it will convert pyruvate to ethanol. As you can see, in a eukaryotic cell, pyruvate enters via active transport. That means energy is used through the membrane protein into mitochondria and is getting converted to acetyl CoA. You need not memorize this molecular formula for the test, but you can see the first carbon dioxide has been released, and NADH, only one NADH is made, guys, not two. This is just indicating what are the different reactions happen during pyruvate oxidation step. Pyruvate is quickly converted to acetyl CoA, but during this process, electrons are stripped off and given to NAD plus, and NADH is made, and one carbon dioxide is released. No, oxygen was not used here, right? No, water molecule was not made. Am I right? Yes. So, glycolysis is common to both fermentation and cellular respiration. Doesn't matter whether the cell is carrying out aerobic respiration or anaerobic respiration or fermentation. Glycolysis is must. Whatever 2 ATP come out of glycolysis, they are used by cells for surviving. So, what is citric acid cycle? Oh, let me go back. This is the drawing that I would like you to draw for learning more and for extra credit. Did you notice that one glucose undergoes glycolysis, and only one glycolysis, right? One glucose undergoes glycolysis and it will make two pyruvates. So when it comes to pyruvate oxidation, each pyruvate will undergo oxidation separately. So whatever pyruvate forms, it will form acetyl CoA. So if it is one glucose, then two pyruvates, so there will be two acetyl CoAs. So yes, there will be two NADHs total and two carbon dioxides. Try to understand this. If the question asks per glucose how many acetyl CoAs are formed at the end of pyruvate oxidation, then you have to say it is two, because there are two pyruvates, right? They double up. Coming to citric acid cycle, yes, this is going to continue in mitochondria in eukaryotic cells. What happens in citric acid cycle is each pyruvate is going to undergo all the chemical reactions which are there in this chart, as you can see, and is converted into or broken down into carbon dioxides. And this picture you might see on the test, and what you will see is the question says something like, what are all the inputs of step number 3? So I would say isocitrate and NAD plus. And if they ask you what are all the outputs of step number 3, I would say carbon dioxide, NADH, H plus, and alpha ketoglutarate. So the simple fact that whatever is at the beginning of the arrow, that is input. Whatever is at the tip of the arrowhead, that is output, right? So you don't have to draw this chart. What you have to draw for extra credit is this one. And you don't have to draw this entire thing. What I want you to draw is from here to here, this part. And yes, all of this should be written here. Okay. My battery is running out. So, well, some things that you need more from citric acid cycle part for understanding the concept better is that before citric acid cycle there was pyruvate oxidation. So per glucose there were 2 acetyl CoAs, and each acetyl CoA is going to undergo citric acid cycles. So 1 acetyl CoA, 1 citric acid cycle, 2 acetyl CoA, so there will be 2 separate citric acid cycles. In this process, the product from breaking down of glucose, whatever has formed, gradually is going to further break down and finally become carbon dioxides. During this process, do we have any profits? Yes, very much. Per citric acid, it is 1 ATP, 3 NADH, and 1 FADH2. Now, what is this FADH2? This is a cousin of NADH. Again, another electron carrier. That's it. Before I move on to other stages of cellular respiration, I want you to learn and remember that mitochondria has several parts, and you have to understand that pyruvate oxidation, citric acid cycle are taking place in the mitochondrial matrix, right? Mitochondrial matrix. That's the fluid in the mitochondria inside the inner membrane, and you see these inner membrane folds. These are called cristae. These inner membrane folds, they have tiny proteins embedded in them, and these are electron transport chain proteins. So yes, oxidative phosphorylation is going to take the help of these electron transport chain proteins. So it occurs through the membrane proteins of the inner membrane of the mitochondria. The space between the outer membrane and the inner membrane is called intermembrane space. This space is very important. I'll let you know what role it plays in coming slides. So just remember, pyruvate oxidation and citric acid cycle take place in the mitochondrial matrix, and electron transport chain occurs through the membrane proteins in the inner membrane, and also the intermembrane space, the space between outer membrane and the inner membrane, is also required for oxidative phosphorylation. Moving on. A little more notes for you to read about try to memorize the inputs and outputs. Hey guys, remember that if they say per citric acid cycle how many ATP are formed, you'll say 1. If they ask you per glucose how many ATPs are formed till citric acid cycle, then you have to add up everything that have formed. Glycolysis is only 2 ATP. There were no ATPs formed during pyruvate oxidation. So per citric acid it is citric acid cycle, it is 1 ATP. So it is going to be 2 ATPs per glucose. So till the point of citric acid cycle, number of ATPs formed per glucose will be 4. 2 during glycolysis, 1 per citric acid cycle. There are 2 citric acid cycles because there are 2 pyruvates formed, each one undergoing citric acid cycle. So there will be 2 per glucose. Till citric acid cycle, there are a total of 4 ATPs formed. But we thought that there will be 32 ATP, right? When will they be formed? There are more 28 ATP to be formed. How are they going to be formed? Because glucose has been broken down completely into carbon dioxides and waters by this time. The thing is, there is another stage called oxidative phosphorylation. But the question again is, how is it form ATPs when there is no more product from glycolysis? All of them, glucose has been broken down into carbon dioxide, right? So let's see. Oxidative phosphorylation is a way to make ATP. ATP can be formed in 2 different ways. One is called substrate level phosphorylation, the other one is called oxidative phosphorylation. So let's see what is substrate level phosphorylation. When an organic molecule has a phosphate attached to it and ADP is floating around in a cell, this is an enzyme with 2 active sites. One active site for the organic molecule which has a phosphate, another active site is for ADP. So what this enzyme does is, because it has the active sites which are complementary in shape to these molecules, one is organic phosphate molecule and organic molecule with phosphate, and another one is ADP. The enzyme will trap both, then it will snuggle and bring them close together so that phosphate will be added to ADP. That is how ATP is formed, and once they have formed, they change the shape and they fall off. So if phosphate is removed from the substrate and added to ADP, and that is how ATP is formed, this is called substrate level for phosphorylation, because the substrate has a phosphate. And ATPs that are formed during glycolysis and citric acid cycle are by substrate level phosphorylation. Then what is oxidative phosphorylation, right? So answer this question is quite obvious. What is oxidative phosphorylation? Oxidative phosphorylation is the last stage of cellular respiration, wherein the ATPs are formed by adding inorganic phosphate, not the phosphate that is attached to organic molecule, but inorganic phosphate that is not attached to any organic molecule. That is called inorganic phosphate will be added to the ADP molecule, and that is how ATP is formed. So try to finish this chart. You don't have to turn in this extra credit to me because this is something that you need for test and understanding the concepts. So what is oxidative phosphorylation, which is making of ATP by adding ADP to inorganic phosphate? What is this stage going to use? It is going to use the electron carriers, NADHs and FADH2s that were formed during all these previous stages: citric acid cycle, pyruvate oxidation, glycolysis. Remember, they all made lots of NADHs and FADH2s. If you can count all of them together, you must see that there are a total of 10 NADHs and 2 FADH2s that are formed. Guys, remember you have to double up everything that is going to be formed during pyruvate oxidation and citric acid cycle. So these NADHs and FADH2s, now they are going to serve as inputs for oxidative phosphorylation. So let's see what oxidative phosphorylation is. Oxidative phosphorylation is the last stage of cellular respiration, which takes place inside the mitochondria after citric acid cycle, and it is going to use all the NADHs and FADH2s that are formed from the previous stages of cellular respiration. Have you guys been to Chuck E. Cheese? You might have seen there that the game machines there, they do not allow your quarter or dimes or pennies in their machines. If you put them also, it will just pass through or it will get stuck. You cannot play the games using them. Just like that, the proteins and the molecules in mitochondria are unable to use glucose or the products of glucose for making ATPs. What they recognize and what they can use are the NADHs and FADH2s. Just like Chuck E. Cheese uses Chuck E. Cheese emblem coins. So these proteins and enzymes which are in the mitochondria, which carry out oxidative phosphorylation to make lots of ATPs, they recognize only NADH and FADH2s. They take the electrons from these electron carriers only. Though food molecule has electrons, they won't take electrons from them. Just like Chuck E. Cheese uses Chuck E. Cheese game machines can use only Chuck E. Cheese emblem coins, not the quarters or pennies or gold coins. So oxidative phosphorylation, as you can see, NADH has been floating around. It bumped into the one of the protein here in the inner membrane of the mitochondria, and because this enzyme complex 1 is greedy for electrons, it is going to grab away the electrons, in fact snatch away the electrons from NADH. And this quinone, which is situated very next to it, is not going to keep quiet. As soon as enzyme complex 1 gets the electron, it will just grab away from it, because quinone is more greedier than enzyme complex 1. Guess what? Enzyme complex 2 is even more greedier. So as soon as it bumps into enzyme complex 3, it is going to simply take away that electron. And cytochrome C is going to take that electron away. And enzyme complex 4, being more greedy than cytochrome C, is going to take away the electron. And finally, oxygen, being the most greediest one, is going to snatch the electron away from enzyme complex 4. So this path of electrons from NADH finally to oxygen, this is called electron transport chain. Where is it happening? It is happening in the mitochondria. Where are these proteins located? These proteins are located in the inner membrane of the mitochondria. So electron transport chain is carried out by the proteins of inner membrane of the mitochondria. Did you notice one thing? That when electrons are jumping, they're releasing energy, and that energy is used by these enzyme complex molecules 1 and 3 to push the hydrogen ions from mitochondrial matrix through them into the intermembrane space. This green this green area is intermembrane space. So as you can see, when electrons are jumping, they're releasing energy. That released energy is activating these enzyme complex molecules to pump hydrogen ions from inside the mitochondrial matrix to the intermembrane space. These hydrogen ions, when they are pumped out, they are going to get collected in this intermembrane space. You have to understand one thing, that when electron is jumping from one protein to another protein to another protein, each one is undergoing reduction first and then oxidation. So quinone gets the electron, it undergoes reduction, and then it will give away, so it will undergo reduction. So these are redox reactions which are taking place. So electron transport chain is full of redox reactions. Finally, oxygen is gaining the electrons. So oxygen undergoes only and only reduction. Oxygen is undergoing reduction because it is gaining electrons. So it is going to form water after undergoing reduction. and oxygen is undergoing reduction, so it must be an oxidizing agent. NADH will lose electrons, so NADH will only undergo oxidation. So NADH is a reducing agent. It is undergoing oxidation, so reducing agent. Oxygen undergoing reduction, so it is an oxidizing agent. Okay, and remember that there's something happening when these electrons are being transported from one protein to another protein, or one molecule to another molecule. That is, these proteins are pumping out hydrogens. It is called pumping. It is not diffusion or just movement, because energy is used to push them out. Energy coming from electrons that are jumping from less electronegative to more electronegative molecule. That process is causing this movement of hydrogen ions. So this is pumping of hydrogen ions from where to where? From mitochondrial matrix to intermembrane space. So electron transport chain, which is a substage of oxidative phosphorylation, is movement of electrons from NADH through all these molecules and proteins in the inner membrane of the mitochondria, and finally ending up with water, or accepted by oxygen and becoming water. So they will be added finally to the water molecule. This is electron transport chain. Was any ATP made during this process? No, there was no any ATP made. Then what is made? Water molecules are made. What are being used up? NADHs and FADH2s. 2 FADH2s and 10 NADH from each glucose molecule. All these are used. No ATP formed. But then we thought that oxidative phosphorylation is going to make lots of ATPs, right? Where are they going to be formed? Let's go there in a minute. But before that, let's understand what has occurred. We have collected, or mitochondria have collected, lot of hydrogen ions in this intermembrane space. Remember, hydrogen ions are H plus. They have a positive charge. Membrane is made of lipids, so lipids do not allow anything that has charge to pass through them. So they are stuck. The outer membrane will not let these hydrogen ions to go out of mitochondria. Can they go back in? They can't go back in, because these are one-way streets. Just like your downtown streets are one way, these enzyme complex molecules can only push hydrogen ions from matrix to outside. They will not let them go back in. But then mitochondria is going to burst if those hydrogen ions are not distributed, or they are not going to pass out. They have to go somewhere else. Well, this collection of hydrogen ions here, this gradient of hydrogen ions, so you know that this is hydrogen ions, so it is acidic, right? So this acidic environment or collection of hydrogen ions is referred to as proton motive force. This proton motive force is going to give energy to the cell to make ATPs. How are they going to be made? So the second substage of oxidative phosphorylation is chemiosmosis. All these hydrogen ions, they are getting suffocated, stuck between the two membranes, inner and outer membrane of the mitochondria. These electron transport chain proteins and molecules, they are not letting them go back in. So there is a nice one next to those little molecules. There is one nice molecule. There are many of them, in fact. These nice molecules are called ATP synthase. As you can see in the inner membrane of the mitochondria, there are many ATP synthases. What they do is they provide a channel for hydrogen ions to pass through back into mitochondrial matrix. And when they pass through, they're going to rotate this part of the ATP synthase, which is going to join ADP with phosphate and make ATP. So that churning of that rotor part of the ATP synthase molecule, due to the flow of hydrogen ions, which is called proton motive force, is going to provide energy to add ADP to phosphate. So this ATP synthase is an enzyme molecule associated with hydrogen ion channel, and it is going to synthesize ATP by adding ADP to phosphate. It is not going to synthesize ATP from food. It is just regenerating ATP. Just remember that. So this process of flowing or diffusion of hydrogen ions through ATP synthase is called chemiosmosis. One more time. Chemiosmosis is diffusion of hydrogen ions through ATP synthase from intermembrane space of mitochondria through ATP synthase to mitochondrial matrix. This is called chemiosmosis. Chemiosmosis, which is substage of oxidative phosphorylation, will make or regenerate ATP. This is a very good video. I would like you to watch this video whenever possible and understand the process of ATP synthesis during chemiosmosis even more. This is the diagram that you need to draw for understanding the concept of oxidative phosphorylation. Everything, this entire thing, not only till here, this entire thing, make sure that you draw all this for your extra credit. You have to follow and understand the red arrows, which is pumping of hydrogen ions and diffusion of hydrogen ions. This is, guys, pumping. This is diffusion. Remember, energy is used for this, whereas energy is not used for this. This is happening just because, you know, there are more hydrogen ions on the outside than the inside, so they are just flowing through. This is diffusion. But diffusion of hydrogen ions is making ATP, and this yellow path is the path of electrons. This is another good animation for you to watch and learn what happens during electron transport chain of oxidative phosphorylation. NADH bumps into enzyme complex 1, electrons are stripped off and taken, but then cytochrome complex C will take the electrons away, and when electrons jump, energy is released, which is used by these enzyme complex molecules to pump hydrogen ions from matrix to intermembrane space. And finally, the electrons have lost a lot of energy, but then oxygen is greedy for electrons, so they are going to take the electrons. Try to answer these true or false questions. You have to remember this path of electrons and energy. This is important for the test. Let's see what is anaerobic respiration and fermentation. Before that, you have to remember that you need all of this for the test. Yes, you have to remember all this for the test, all these numbers. Can we break down food without oxygen? Yes, life is possible without oxygen. Do we make ATP? Yes, very much. Can we oxidize without oxygen? Yes. Instead of oxygen, we will use sulfate. Not we, but sulfate or something else. Bacteria do use. What is the role of oxygen in cellular respiration? They are the final electron acceptors, not donors. They are the final electron acceptors at the end of electron transport chain. Remember that. So as I said, in anaerobic respiration also there is glycolysis, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, which has both electron transport chain and chemiosmosis. Only thing is, finally, instead of oxygen accepting the electrons which are jumping from NADH to all those proteins, finally there will be sulfate or nitrate or carbonate which is accepting those electrons. So instead of water, if sulfate is there to accept the electrons, then it will form H2S, hydrogen sulfide, and that is what is happening in the bacteria in the lakes, and where, you know, the marshes, whenever you drive by, sometimes your car suddenly starts to stink. That's because of the bacteria in the lakes. They are carrying out anaerobic respiration, not using oxygen, because they are stuffed inside the water and in the mud. So they do not have access to oxygen, so they have adapted themselves to use sulfate, and instead of releasing water like we do, they are making the product called hydrogen sulfide, which gives that rotten egg smell, and that is why your car suddenly starts to stink whenever you're driving by the lake. There are two terms that you need to remember. Obligate anaerobes means they use anything other than oxygen. They do not want to use oxygen. They're so strict that, you know, if you give them oxygen, there's a bacteria called Clostridium botulinum. You simply give it oxygen, it will die. And they are called obligate anaerobes. Same thing, obligate aerobes. They are so strictly oxygen dependent that, you know, if they don't have oxygen, they die. Like we people, we can survive for some time, but not very long, because, you know, our brains need oxygen. We faint first. Our heart is still pumping, so we can be revived. But our brain, which cannot carry out fermentation in the absence of oxygen, it will lose its ability to function, because there is no ATP. What is facultative anaerobe? Facultative anaerobe is the one which is ready to adjust whether there is oxygen or no oxygen. It will still keep keep on making ATPs. So they survive both in the presence or absence of oxygen. Example is our muscle cells and yeast cells. What is fermentation? It is partial degradation of a food molecule because there is no oxygen. So they simply partially degrade food molecule and produce ATP through glycolysis. Yes, they have NAD plus using as the electron carrier. So remember, in fermentation, first glycolysis occurs, and whatever the product is formed, which is pyruvate, always, pyruvate is going to undergo another process to release NADHs, because if there is no NAD plus, remember, glycolysis cannot occur, right? So the purpose of fermentation is actually converting pyruvate into something else and take the electrons away from NADH. That is how the process of glycolysis will continue, and the organism or muscle cells will be able to make a lot of ATPs through a lot of glycolysis. So not that many per glycolysis, only 2, but something is better than nothing, right? Exactly. Same thing happens in lactic acid fermentation, which takes place in our muscle cells. Glycolysis occurs, 2 ATP, whatever are formed, they are going to serve our energy requirements, and pyruvate is converted into lactate, which is the ionic form, or lactic acid, which is the non-ionic form. And pyruvate, when it is converted to lactate, it will take the electrons away from NADH and will free up some NAD plus, so that the process of glycolysis can go on. So what is alcohol fermentation? It's actually a mode of releasing energy for bacteria and surviving. The bacteria and the yeast are not making wine because they know that, you know, people require alcohol. Oh my God, let me get to work. No, it's not that. Yeast want to survive. You give them food, you know, fruits or flour, bread flour, right? And you provide water, and it is happy to eat and divide and make more of itself. This is yeast cell under the electron microscope. Showing this process, remember, carbon dioxide is released. That is what is fluffing up the bread, and the bubbles that are formed in wine or champagne actually are the ones which are added on top. That is not coming from the fermentation. That is what I read in the scientific articles. And the purpose of fermentation is always to release and free up some NAD plus, so that the glycolysis process can continue. Okay. Same thing with our muscle cells. They carry out glycolysis to give energy to the muscles that we are working out, and we can use lactic acid fermentation for making cheese and yogurt also. The pain that is caused in our muscle after workout is not actually due to the lactic acid um, uh, accumulation, but it is because of the high levels of potassium, and lactic acid is blamed unnecessarily. And don't confuse this lactate with lactation. Lactation, that milk, that is something else. Lactate is another word for lactic acid. Lactate is ionic form of lactic acid. So only our muscle cells carry out lactic acid fermentation, which is breaking down of food to release energy and continue the process. Purpose of fermentation is to free up NADH to make NAD plus, so that glycolysis can continue. Yes, both process, fermentation, cellular respiration, and including aerobic and anaerobic respiration, all of them use glycolysis, all of them. Different types of food. We not only eat proteins, carbohydrates, fats. We use different types of carbohydrates, polymers, such as not glycogen, starch, and disaccharides such as maltose, sucrose, lactose, right? We use different types of proteins. When we eat burgers, we are actually eating the myosin and actin proteins, which are in the muscle cells of chicken and fish. I'm sorry to say that, but yes, that is what we are eating. So when you eat, let's say sucrose, which is made of glucose and another sugar, then glucose will start the journey from here on, whereas another sugar is going to join somewhere in the middle. When amino acids, which are coming from proteins, are provided to a cell, then they will join their or when they will join the cellular respiration pathway, either forming into a pyruvate or acetyl CoA, or one of the products of citric acid cycle. Okay, so remember that fact that whatever we eat is not getting converted to glucose. It will join somewhere in the cellular respiration pathway, and it is finally oxidized if you have oxygen, or will undergo changes in order to give you energy through cellular respiration pathway. If you eat extra, then it will be converted to fat. Even extra protein is not good. We have to eat only 1 gram of protein per kg of your body. Whatever extra protein you eat, it is going to be a burden on kidneys. So extra protein is not good. Extra sugar is not good either. How is cellular respiration regulated? Cellular respiration is regulated by feedback inhibition or negative feedback method. Cellular respiration can be regulated at a couple of places. One is phosphofructokinase. which is an enzyme which is converting the products during the glycolysis step. That is fructose 6 phosphate to fructose 1,6 bisphosphate. This enzyme is regulated and is stopped from converting fructose 6 phosphate to fructose 6 bisphosphate, 1,6 bisphosphate. When you have a lot of ATPs. ATPs will stop them from working by changing its active site shape. This is called non-competitive inhibitor. Citrate also can do the same thing. Can you imagine how citric acid cycle, which is forming the citrate in its pathway, using the acetyl CoA coming from pyruvate oxidation, which is taking place in mitochondria, is going to stop the process or the enzyme which is working in the cytosol? By diffusion. Citrate is going to get accumulated, lot of it in the mitochondria. If there is no oxygen and cell is not using up, then citrate is going to come out and bind to phosphofructokinase and going to stop this pathway here. If this stops, and the chain of sequential events cannot take place. So phosphofructokinase, which is an enzyme in glycolysis, is going to be inhibited by ATP or a citrate, not and, or either this or that will stop the pathway early in the process. These are the products of cellular respiration, hence it is called feedback inhibition or negative feedback. Make sure that you know the answers to all these questions and understand the concept. Play and replay the video and make lots of notes. Take advantage of the source provided to you, and good luck.